So we've started down a trip through some of the lines of evidence for how evolution has happened. Quick review that fossils are real and a thing that happens sometimes if you're super lucky, if you die in exactly the right place and you got the hard parts for it, and the hard parts get replaced by the right minerals, maybe your hard parts will be found by a 10-year-old 80 million years later. Or, then there's the really lucky ones that like get frozen or sunk into a tar pit. Hazen?
Yeah.
What? I don't trust you.
We should talk about how I love fossils, and this is very interesting.
Ohio is like a freaking hotspot for fossils. Like, we have tons of Ordovician fossils.
Yeah, imagine, I can't wait to go to Ohio.
Right. Yeah, it's weird that we're known for anything, but yeah, as they think, like from a particular period, we are just stinking with fossils. Ordovician, which is a particular period in history. A lot of horn corals, trilobites, what do you call it? Brachiopods. Yeah, it's all ocean stuff. Cedar Creek is, yep, it's awesome. You're allowed to take anything smaller than your fist.
We went there in like second or third.
Yeah. What were you gonna say, Brady?
So short. Date is the hero capital.
Number one,
That is strong.
yeah, number one.
Anyway, you might be tempted to think that fossils are the only way we know how stuff was back then. They're not. So let's talk about that today. First of all, what we call homologous structures are a big line of evidence we use to see kind of what's related to what and what's evolved from what. Basically, homologous structures, not to be confused with homologous chromosomes, which remember are those chromosomes that have the same genes but different alleles. These are body structures in different species that, while they look somewhat different in the adult, come from the same blocks of tissue in the embryo, and a lot of times are hard to tell apart in the embryo. So, for example, this is a bat's wing under the hood, right? That's the bones that underlie a bat's wing. This is a mouse front paw, and this is a human arm. Real different in the adult forms, right? You can fly with that one. You can do whatever mice do with that one. Hold cheese. What do mice do? You should probably know that. And you can play piano with that one. Very different in the adult forms. But if you look at them in an embryo of a mouse, a bat, and a human, you'd be hard pressed to tell them apart. They're coming from exactly the same spot. It's basically the same parts list, same number of bones. And so basically, homologous structures tell us things about who's related to whom. For example, that mice, bats, and humans, if you didn't know already, are all mammals, which means they all have a common ancestor. Vertebrates are fun for homologous structures because basically we've all got them to some degree or another. So you can widen this out and say, all right, let's look at all these things. Let's look at stuff that's adapted for flying, swimming, running, and grasping. We've got the same parts list, greatly sort of shaped and adapted based on environment, which brings us to the fact that some things look alike not because of common ancestry. Specifically, analogous structures sometimes we see critters that look alike, that have similar features, for a totally different reason. Let me give you an example. I'm about to show you a bird, a fish, and a mammal. That have some pretty similar structures. Giving some structures these things have in common.
Fins.
Yeah, they've all got fins. Not only do they all have fins, they all have certain fins in the same spots, right? Like that one, and that one. Those are pretty much positioned the same way.
They all have white bellies.
Two of them definitely have white bellies. I'm not sure about this one. Why do you think that is? What do you think the white belly's for? Landon?
I don't know, but
What do you think the white belly's for?
I think it's because when you look down, you can't see them. What I mean by that, like when you look, if you're the water surface and you look down into the water, you can't see them. It's like camouflage for them.
Why? Because they're belly up?
No, the top's like, you see, there's.
That's why the top's dark, you're saying.
Yeah, so.
Why is the belly white?
Like the more dangerous fishes look up, they normally just see completely white.
Because the top of the water often looks white. Absolutely. It's camouflage both ways. You're right. Yeah. And so at least two of them have that. Landon?
they all have eyes.
They do all have eyes. That's true. Two eyes.
They all have.
Eyes we can open under the water.
Okay. Good enough. Yeah.
Their heads come to like a point.
Ooh, not just their heads, but like they kind of have the same body shape, don't they? How you want to describe that body shape?
Football.
You guys are spicy this morning. Yeah, football, torpedo maybe shape, teardrop kind of. So what I'm going to tell you is, it's not because they're related. I mean, everything's related, but these similarities are not because they're related. Why are they? Why do they all have torpedo shape and fins in certain spots and, white and dark in the same way? Chris? Okay.
Environmental factors.
Yeah, they live in the same environment, don't they? They all live in the ocean. It turns out that is one of the prime ways to be awesome in the ocean, to have fins and a torpedo shape and a flat tail. And if you're a predator, camouflage. I suppose if you're a prey as well. So analogous structures are similar, not because of common ancestors, but because of shared environments. And the kind of evolution that results in two different species that are distantly related ending up looking similar, that kind of evolution is called convergent evolution. Converge means to come together, right? So, throw two different species in a very similar environment, often you will see they evolve very similar characteristics. Not because they're related, but because that's a way to be awesome in. So homologous because of common ancestry, analogous because of convergent evolution. So you can see both of those are sort of ways to track what's happened to species throughout evolutionary history. Yeah.
Do they have that thing on top?
Why do they have that fin on top? Anybody want to tackle it?
In it because aerodynamics and also
The air.
Hydrodynamics. Hydrodynamics, yes.
yeah, and if they turn you're
So, you ever seen a surfboard?
Yeah.
The fin on the back?
Yeah.
Same thing. It keeps you from slipping from side to side, keeps you going in a straight line. Questions so far? Homologous, analogous. Then, here's my favorite kind of structure that tells us stuff about evolution. vestigial structures, which are hilarious and ridiculous, but very informative. How many, raise your hand if you've had your appendix taken out?
My brother.
He's fine, isn't he? You know why? Because you don't need your appendix. It's just decorative. No, what it is a leftover. It's an evolutionary leftover. It's basically, if we wind up being able to dissect this year, you'll see one in an animal that can use it. You'll see, if you dissect a rat, you'll see the rat has this big old blind pouch coming off of its large intestine called a cecum. And what happens is rats eat just gross garbage. And when they eat stuff that's really hard to digest, like, teeth or nails or something like that, goes right into the cecum and it hangs out for like weeks and softens up so that eventually it can pass through the system. That's what it's for. It's just that humans don't eat garbage anymore. We don't eat like an entire vertebrate animal in one gulp. And so we don't have to be able to digest, bones and hair and stuff. And so gradually the appendix has evolved to be smaller and smaller. And now it's just this little pinky-sized thing hanging off the corner of your large intestine. That's a vestigial structure. It tells you stuff about what your ancestors did, right? That's the idea. Ever seen a whale skeleton? If you ever go to Washington, D.C., and you get to go to the Smithsonian Museum of Natural History, did you see the whale skeletons? Oh my God, they're so cool. When I went there, I didn't know they were there. And I went there like, I don't know, 6 or 7 years ago, and they're just hanging above you. It's this right here. It's like these four things hanging above you. Like I said, sometimes we luck out and we get the fossils of the things that are in between the stages of evolution. So like this is a recent whale ancestor right here. This is Pakicetus, which is a direct ancestor of whales. And these are kind of our in-between phases. And what you can see is the legs go away because it turned out to be easier if you just hang out in the water all the time not to have legs. But the thing that didn't go away, because it must not have been a disadvantage, is the pelvic bone, the hip bone. It's still there in modern whales. Like, it's just floating there, not connected to anything. That's a vestigial structure. It's not doing anything. It's not hurting the whales, but it tells us something, right? It tells us that whale ancestors must have walked on land. Homologous, common ancestry. Analogous, similar environments. Vestigial, ridiculous. Leftover, but tell us something.
Have you had your appendix removed?
I have not. Not.
You can just eat rocks.
Well, I think that would probably result in having to get mine. I don't know. But yeah, it's a big deal when the appendix gets something stuck in it, it gets infected. Yeah, like it could kill you.
It literally has one.
It has no chance. All right, last thing. You don't have to draw that because you're about to do it on your assignment.
Wait, but Mr. Sandros, you already have something.
Yeah, you're gonna get time on that too. How is it? Oh yeah, good. There's plenty of time. Now that we can read DNA, and now that we know what proteins are made of, and we know that the proteins are the way they are because the DNA was the way it was, and we know that DNA changes gradually over time, descent with modification, there's that thing where we can basically measure how similar two organisms are genetically and figure out when the last common ancestor was. Well, rather than just measure all the DNA, the thing you're going to do today, you're going to pick one protein that all the critters have in common and count how many amino acids are different from one critter's protein to the other critter's protein. That way you don't have to measure the whole set of DNA. Pretty slick. And the one we're gonna pick, let me make sure it's that same one. Yeah, cytochrome C. Remember cell respiration? Remember that nightmare of a chapter? Remember the electron transport chain? The ding, 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 ding of the electrons? One of the ding things, one of the proteins that grabs an electron and passes it on is cytochrome C. And every living thing that does cell respiration has to make cytochrome C. But because a lot of them have been separated from their common ancestor for a really long time, their cytochrome C has mutated. Like, you're just going to be counting those mutations. And by counting those mutations, you're going to be able to tell that the ones that have more differences between their cytochrome C, it's been longer since they had their common ancestor. In fact, a lot of what we're going to do for the next few weeks is going to be about establishing relationships like that by measuring differences. So let's give it a shot. Genes are big. You know that, right? Like proteins are long, lots of amino acids. So when you get this, turn it to the back page to see just how long cytochrome C genes can be. Oh boy. Now what you're looking at isn't actually the gene, it's the protein. So let's all look on the back. Notice that you're looking at, these are not all different critters. This is 2 rows of the same critters. So this gene is that long, not the gene, this protein is that long. Cytochrome C, each letter in the row is a different amino acid. And the key to the amino acids is in the lower right, and you don't have to worry about that key at all. That key does not matter to you. You don't care which amino acids are isoleucine and histidine. You don't know what that means. Those are just the 20 different amino acids. What you got to figure out is where the differences are. That's all. You're just looking down these columns and circling any differences you find. Not yet, because you don't have to do it for all of them. You just have to do it. For starters, if you look on the front page, it's asking you, what is the number of amino acid differences between a human and 5 different other animals? So you can limit your search to that. Just find the human rows and the rows for the other things. Make sure you look at both rows, right? Because human is here and here. That's the whole gene. But once you find all the differences between human and rhesus monkey, you can move on to kangaroo, and then snapping turtle, and then bullfrog, and then tuna. Then you can turn the page and start to, don't miss the little hexagons on the second page. That's where you're going to end up writing those numbers of differences. Okay, but that's called a cladogram, and we're going to spend a lot of time doing those in the next couple of weeks. All right, that is your assignment. Get done with that. You can work on your Edpuzzle, if you like. Let's make this one due Monday. Yeah, this one's due Monday. So I guess if you would rather work on your Edpuzzle, you could do that.